Good morning, everyone. Welcome to the Alliance for Regenerative Medicine's 2022 Cell and Gene Medicine State of the Industry Report and Sector Panel Discussions. I'm Emile Nuwaysir. I'm the chairman of ARM. I'm also the CEO of Ensoma and the chair of BlueRock Therapeutics. I'll give you a brief set of introductory remarks, and then I'll be joined by my colleague, Janet Lambert, the CEO of the Alliance, and she'll give you an overview of the state of the sector as of the end of 2021. And following her industry report, we have the pleasure of two different panel discussions for what we should expect in 2022 from some of the key executives in our industry. But before I pass the baton to Janet, if I may exert chairman's prerogative for a minute to look back on some of the remarkable science that we've performed last year in the sector and just how a few of those experiments have fundamentally altered our perspective of what's possible or how soon we might realize the promise of regenerative medicine. So if I may, thank you. Let's start with CAR-T, which for years has held the promise of a broad transformation in oncology. But to date, it's been limited to use when the standard of care has failed or when other treatment options have been exhausted. You know, it's been a fourth or a fifth line treatment. And that is changing. This year, we saw Gilead and Bristol Myers Squibb release data showing that their CAR-T therapies, Yescarta and Breyanzi, respectively, improved event-free survival over the standard of care second-line treatment. And those same therapies are showing suggestions of even more promising first-line treatments. So this advancement in the field has been primarily because we've learned how to handle the safety of these incredibly potent medicines. We've learned how to utilize a living cell as a medicine. And that type of learning applies to all the cells that follow, to the entire field of immuno-oncology. So it's very exciting. And this would lead to a reasonable scientist to conclude that very soon CAR-Ts will be a first-line therapy. But these therapies are based on autologous sources, and we all know this will limit the accessibility. So many have pursued gene editing strategies to convert allogeneic sources into cells with reduced immunogenicity. And the open question was, how would these editing strategies, would they yield potent cells, and how would these allo cells compare? And studies now suggest last year that the initial responses to these allo-edited CAR-T cells are in line with the responses of the approved autologous CAR-T therapies. The complete responses roughly match the CRs from the auto cells. And most notably was a result from CRISPR Therapeutics last year, where the potency appears to be pretty good. So the open question remains of durability of these new medicines, and that'll take some time to learn. But nevertheless, this is a big step for the field. We can allow ourselves to be optimistic that future patients will not be shackled to this autologous paradigm. Will we next year be talking about allo-edited cells proving superior to auto cells for liquid tumors? That'll be fun to ask. Another paradigm, which we've changed this year, was that in vivo editing directly in the human body is on the distant horizon. It's not near term. But we've had some compelling results this year that demonstrate the horizon is closer than we thought or might have hoped. Intellia Therapeutics released their first data from an in vivo CRISPR trial last summer, which showed the therapy could be an effective treatment for patients with a rare genetic liver disease. And there are many other editing platforms with similar, very compelling preclinical data and with a broad range of technologies and new approaches. And this leads you to think, for example, in the oncology field, where the intersection of in vivo editing with all the advances in CAR-T engineering will lead us to a future where, at least in some cases, we eliminate entirely the ex vivo cell manufacturing paradigm. Whether it's allo or auto, it'll become unnecessary. It's incredibly exciting. Another paradigm we've challenged was that genetic medicines rely on changing the code itself, the DNA itself. But this year in particular, we've seen an explosion of new ideas on how changing the memory, the genome memory, might be able to cure the disease. Methods like epigenome editing, where we change the DNA methylation status, or where we squeeze the chromatin to alter gene expression, or RNA edit. Companies like Chroma and Tune Therapeutics made headlines towards the end of the year with the launch of their epigenome platforms. Another constraint that we've been challenged by is that our treatments, while incredibly powerful, can't reverse the damage that's already occurred. There are, in fact, hundreds of millions of people that suffer from degenerative diseases, and the conventional wisdom is that their degenerative journey is a one-way street. Our therapies can only prevent further damage. This would be true for degeneration from simple genetic disease as well as complex chronic disease. But this year, we have notable examples where that is not true, where, for example, a limited trial in AADC deficiency, which is a severe inherited neurological disorder, suggests that an 8-year-old treated with the gene therapy was able to regain neurologic function after treatment. So maybe the body is more plastic than we thought. But in most cases, this degeneration, especially in complex polygenic disease like diabetes or Parkinson's, is probably irreversible, right? And the cell is lost. Once the degeneration is severe, we can't reverse it. And in my opinion, the most provocative result we saw this year was, in principle, that degenerative disease is reversible. Vertex showed in a single patient at this point that they could restore physiologic glucose control in a type 1 diabetes patient by implanting stem cell-derived beta cells. And this showed that you could replace the lost cells, restore the function, and reverse at least the symptoms of the degenerative condition. That's a profound step, and it's incredibly exciting. Very early data, but incredibly exciting. So next slide, please. So that wraps up what I'm going to say. I mean, just as this, the new telescope, the James Webb telescope launched last month, is powerful, and it allows us to look billions of years into the past. We, with these results, are looking forward into the future to a promise that we can just now begin to see as real, to treatments that move earlier and earlier in the patient journey and that prove truly durable, to broadening the accessibility of our science to more and more patients in need, and to safe and powerful medicines that begin to blur the line between prevention and cure. Thank you for your time. At this point, I'd like to hand it back to Janet for the next portion of the morning.
Thanks, Emile. And hello, everyone. I'm Janet Lambert, the CEO of the Alliance for Regenerative Medicine. Thanks for joining us. Emile described some of the really important scientific advances we saw in our field in 2021. And now I'd like to talk about some of the other 2021 sector developments, what we expect to happen in 2022, and how these events are part of what's driving the transition that Emile just mentioned. But first, for those of you who may be unfamiliar with ARM, a little information about who we are and our role in the sector. ARM is the global voice of the sector, that is the gene, cell, tissue, and broader regenerative medicine sector. Our membership includes more than 400 diverse members worldwide, including therapeutic developers, medical and research centers, tool and service providers, and patient organizations. We're the only international advocacy organization specifically focused on advanced therapies, and we work in the US and Europe on behalf of our membership to bring safe and effective medicines to patients by promoting clear and harmonized regulatory pathways, enabling innovative and reimbursement models and patient access, addressing manufacturing barriers, and educating stakeholders, including policymakers, patients, and the media, about the particulars and the immense promise of this space. As Emile described, the sector is creating new paradigms. 2021 had several notable successes, and 2022 is slated to have regulatory decisions on as many as 9 new cell and gene therapies. We, of course, had some setbacks as well as progress in the past year, but those setbacks are partly what's driving us towards these new paradigms. First, I'd like to talk about the investment landscape, the engine that's driving this scientific progress. The investment patterns in 2021 will strongly support the long-term growth and success of our sector, but it was juxtaposed with some painful volatility. We had another record-breaking investment year. The rise of gene editing continued at a strong pace. Venture capital played a central role, pouring huge sums into early-stage companies. And nevertheless, it was obviously a poor year for equity performance. So let's dig into the numbers a little bit. 2021 set another investment record for cell, gene, and tissue, with $23.1 billion raised over the course of the year, a 16% increase from 2020. You can see here from this chart that investment in cell, gene, and tissue has more than tripled over the past 5 years. Breaking it down a little bit, Gene therapy developers raised the most last year with $10.6 billion raised, followed closely by developers active in cell-based immuno-oncology with $10.1 billion raised. Although gene therapy outraised cell-based IO continues to show the strongest growth with a 26% increase in financings year over year. Looking geographically, developers headquartered in the US raised the lion's share of the investment once again, with $18 billion of the total raised in the US, an increase of about 53% year over year. European developers, by contrast, raised $3.3 billion, an 8% decrease from last year. And Asian developers raised $2.2 billion, a 4% decrease from last year. We also want to showcase the rise of gene editing, which Emile noted in his discussion of scientific advances. Companies active in gene editing raised a total of $8.5 billion in 2021, more than 1/3 of the total raised sector-wide. If you drill down a little bit further, companies active in gene editing made up $4.7 billion of the financing in the gene therapy category, or about 45% of the total gene therapy financings. As a percentage basis, that's up from 38% just 3 years ago. Excitement in this subsector is driven by the in vivo CRISPR data from Intellia that Emile described earlier, but also continuing advances in traditional and new editing technologies. Base editing is set to enter the clinic in 2022, and the approval of the first gene-edited therapy could occur in 2023. The record investment in 2021 was largely driven by venture capital, as you can see in this chart, with $9.8 billion raised, a whopping 73% increase year over year. There were also a record number of IPOs in 2021. 26 companies had IPOs, nearly double the previous record of 14. Total IPO funding was $4.8 billion, up 30% year over year. Even while investment rose to new highs, public equities in the sector, especially gene therapy companies, had a rough year, particularly after the first quarter. We see the recent declines in public equity performance as a combination of macro market forces like inflation fears and individual company and sector news, resulting in short-term volatility. It's important to keep in mind that despite this rough 2021, Rmat, an index that reflects performance across all regenerative medicine technologies, still closed out the year at 177% of 2018 levels. So let me turn now to the commercial and clinical landscape of the sector. 2021 was a notable year for new regenerative medicine product approvals. In fact, with 6 new products approved, it was the second best year on record, following the 9 approvals of 2016. It was also the best year on record for approvals of cell-based immuno-oncology products with 3 new CAR-Ts, approved across the US, Europe, and China, including the first approval of a BCMA-targeted CAR-T, which was also the first CAR-T therapy approved for multiple myeloma. Additionally, we saw product approvals for a serious inherited neurological disorder, a rare and historically fatal birth defect, and severe burns. It was a breakout year for FDA's Rmat designation. 3 out of the 4 products approved in the US had Rmat designation. This designation, which Rmat played a leading role in creating, was established in 2016 and is intended to expedite patient access to innovative regenerative medicine therapies. To date, 67 programs have been awarded Rmat designation. Abecma became the first product with Rmat designation to receive FDA approval, followed by Stratagraft and Rethymic. We're currently tracking 2,261 active global clinical trials in regenerative medicine. This includes both industry-sponsored trials and academic and government-sponsored trials. A quick note to say that the clinical trial data here is from Q3, as there's a lag in the reporting of clinical trials to registries, particularly at the end of the year. We will provide year-end data in our upcoming annual report. Looking at some of these specific indications, you can see that cancer is the #1 target for cell and gene therapies by a country mile, with 1,354 ongoing trials. But you can also see that there's significant activity in neurological disorders, diabetes and diabetes-related disorders, cardiovascular, rare genetic disease, and stroke. Of these, there are 1,129 active industry-sponsored regenerative medicine trials globally. And as you can see here, cell-based immuno-oncology makes up the largest portion of this clinical trial landscape, largely thanks to a huge number of phase 1 IO trials. Cell-based IO makes up more than half of all phase 1 industry-sponsored regen med trials, a relatively recent tilt towards IO. If you look at the 143 phase 3 industry trials, that's the dark blue pie slices in the top row, you can see that 44% are in cell therapy, 33% are in gene therapy, and just 22% are in IO. So again, the substantial shift towards cell-based IO across the clinical trial picture is relatively recent. We're also tracking 1,100 academic and government-sponsored trials worldwide, about the same number as industry trials. As a whole, these trials, not surprisingly, tend to be earlier stage with a significantly different technology mix. In academia, we also see a much larger proportion of cell therapy trials and a much lower proportion of gene therapy trials. Based on this rich pipeline, let's shift our attention to 2022 and beyond. And there are two big stories we see looking to 2022 and beyond. The first is that 2022 is shaping up to be a record year for approvals of new gene therapies targeting rare disease. The second is that we have begun in earnest the gradual evolution in focus towards more prevalent diseases with complex drivers. Here's the current outlook for cell, gene, and tissue therapy decisions in 2022, drawn from public reports. As you can see, it could be a really active year with as many as 15 anticipated decisions across the US and Europe, including 9 on never before approved products. In particular, 2022 will be important for gene therapy's traditional target, rare genetic disease. As of now, we're due to have regulatory decisions on 5 new products across the US and Europe from BioMarin, GenSight, PTC, Unicure CSL, and Crystal Bio. We will very likely see a record number of approvals for this product class in 2022. There are also several key data milestones across both rare and more prevalent diseases that will drive headlines this year. We'll see late-stage gene therapy data readouts from Pfizer's hemophilia program and Solid Bio's DMD program. Just this morning, 2-year data was reported for BioMarin's hemophilia A program, setting up an expected BLA filing in the second quarter. We're also expecting to see additional data from Vertex's cell therapy for type 1 diabetes. And undoubtedly, gene editing will continue to expand its impact in the clinic, with the initiation of the first 2 trials utilizing base editing from Beam Therapeutics and Verve, and an additional gene therapy trial from CRISPR and ViaCyte for type 1 diabetes. We'll also see data readouts from Caribou Bio, Intellia, and others. It's reasonable to anticipate that in 2022, the early stage investment picture and equity performance could both align in a positive direction, especially if inflation expectations begin to subside and our data readouts are positive. Looking at the clinical pipeline in 2022 and beyond, we'll gradually see an evolution from rare monogenic diseases and liquid tumors to more prevalent diseases and solid tumor cancers. As you can see in this table, we have many late-stage programs in a variety of cancers. Importantly, the first gene therapy for a prevalent disease could be just a few years out. The oft-quoted 2019 FDA prediction of 10 to 20 approvals by the year 2025 still looks doable, albeit likely at the lower end of that range. This is something Dr. Peter Marks from the FDA has recently reinforced in conversations with us. Of course, key challenges remain for the sector, and ARM and our members are heavily focused on addressing them. On dosing and delivery, while safety concerns weighed down the gene therapy sector last year, there's reason for optimism. Thousands of patients have been safely treated with AAV therapies to date. The FDA has signaled that it doesn't plan on significant regulatory decisions in the area of dosage caps and will work collaboratively with stakeholders on important measurement techniques. And we have exciting research into other delivery vehicles and technologies for genetic medicines ongoing. CMC remains a big challenge for the sector, but it's one the industry is addressing head-on. Expected congressional legislation and PDUFA should both be helpful in bringing about more clarity in regulatory requirements, and ARM continues to be in direct conversation with the FDA on a range of CMC matters, including CMC clarity. In 2021, we completed something called Project Aging, a best practices guide to using quality by design principles in AAV gene therapy manufacture. And that guide is now becoming a widely used workforce development tool. Finally, the areas of policy and reimbursement, where ARM spends a good deal of its time and effort. 2022 will be a big moment in Europe, which is at a risk of falling behind in the globally competitive cell and gene therapy sector. Legislation emanating from the EU pharma strategy will represent the first major review of the pharma landscape in Europe in 20 years. And ARM is working to make sure that ATMPs can thrive in that reset environment. In the United States, we continue to focus on removing barriers to innovative payment models and ensure that policymakers recognize that cell and gene therapy is different than traditional pharma, and that we have to reorient our system to appreciate value, not just focus on cost. The biggest threat, in our view, to regenerative medicine's advance isn't the science. It's a legacy healthcare system that wasn't built for the science. Given this wave of next generation regenerative medicine products poised to change the life of so many patients, I'd like to close with a reminder of why we do what we do. Jayla Turner was born in 2007 and diagnosed with complete DeGeorge syndrome, a congenital abnormality that causes affected infants to be severely immunocompromised. Without treatment, life expectancy for patients like Jayla is only 2 to 3 years. She received the therapy now known as Rthymic in 2008 at age 1, and she is now 14 years old and in the 9th grade. I don't know if any of the rest of you have a 14-year-old, but if you do, we probably can best find Jayla on TikTok or Insta, or listening to Olivia Rodrigo. At least that's the case with my 14-year-old. This Engs event therapy is one of the therapies approved under the RMAT pathway last year. It's a testament to what we've accomplished, an inspiration for what we must still do to reach many, many more patients. With that, I'd like to thank you for joining us and listening. This is the information that you need to access the slides from today's presentation, including additional 2021 data that we didn't have time to include in the presentation today. You can also sign up for ARM's weekly roundup of sector news, the sector vector, via our website, as well as find a number of other publications and presentations. You can also keep track of the sector by following us on social. With that, it's my pleasure to introduce the moderator of the first panel discussion. Bob Smith is a member of the ARM Board of Directors and a member of our executive committee. He's been the Senior Vice President of Pfizer's global gene therapy business since May of 2016. He's been a wonderful colleague to me, and I'm pleased to turn over the mic to him to introduce his panel. Thanks, Bob.
Great. Thank you, Janet. Thanks for that very kind invitation and for that very comprehensive overview of the state of the industry. Obviously, we have a few challenges yet remaining as an industry, but we're really, really excited about the potential for many of these transformational therapies and all of the work that many of our member companies are doing across the sector. I'd like to start by introducing our panelists. In order would be Suma, if you could please introduce yourself.
Thanks, Bob. This is Suma Krishnan. I'm the founder and chief operating officer at Crystal. We founded Crystal in 2016, and fast forward 2022, as Janice just mentioned, we are very excited we announced our phase 3 data from BVAC for a disease called epidermolysis bullosa, and we look forward to filing the BLA in 2022. So it's very exciting times for Crystal Biotech.
Great, thanks, Umer, and thank you for participating in the panel. Looking forward to your perspective and insights. Why don't we go next to Bobby, if you could please introduce yourself.
Thanks, thanks, Bob. Yeah, thank you very much for asking me to be on this panel. Bobby Gaspar. I'm one of the founders and CEO of Orchard Therapeutics. I'm an academic clinician and scientist by training before we founded Orchard and then became CSO and subsequently CEO. I've been in the field for really 30 years, right at the start of gene therapy for severe genetic diseases using gene-modified blood stem cells, and have been involved in a number of academic trials and then in trials within Orchard. And so what we're doing is really trying to potentially cure these monogenic disorders of the immune system, of neurometabolic disorders. We have an approved product called Lambert for the treatment of a very severe neurodegenerative condition, and that's been approved in the EU and is commercially launching there. And we're excited to bring that to the US as well very soon. And then there's a pipeline behind that as well. And we also believe that we can use this HSC gene therapy not just for rare disorders, but also for larger diseases as well, given what we've seen clinically. So I think it's an extremely exciting time to be able to do this and make a really, and I've treated many patients myself with these gene therapies and just seen what an extraordinary impact it can have. And it's an incredibly rewarding thing to be able to do and now to bring to a wider population. So thank you.
Truly great. And thank you, Bobby. Why don't we turn next to Jason?
Sure, thanks, Bob. So hello there. My name is Jason Cole, and I'm also quite honored to be on this panel today. So I'm the Chief Business Officer at Bluebird Bio. That makes me responsible for corporate strategy, corporate development, external affairs, and operations. And so late last year, Bluebird completed a spinoff of our oncology or cell therapy business. And as a result, we are now exclusively focused on ex vivo gene therapies for rare diseases. And we have a number of late-stage gene therapy programs, 2 of which have been filed with the agency and are part of Janet's slide of upcoming potential approvals. And the 3rd of which, our sickle cell program, is also a program we're advancing and hoping to file by the beginning of next year. And like others on this phone and in the industry, we remain committed to research and exploring new areas to apply our technologies.
Thanks, Jason, and congratulations on the completion of the transaction, and looking forward to hearing more about your pending approvals. Why don't we turn next to Harold, if you could please introduce yourself.
Yeah, good morning, Bob. I am the Chief Medical Officer and Head of Clinical Development at BioMarin. I believe we're celebrating our 25th anniversary this year. For those of you who don't know BioMarin well, our R&D strategy has always been that we work in disease spaces where the molecular basis of the condition is well known. We target our interventions at the fundamental molecular defects to create transformative medicines. We look to demonstrate early benefits so that we know we're on the right track using early efficacy signals or biomarkers. And in particular, beyond gene therapy, which we're talking a lot about today, especially at BioMarin, we also have multimodality expertise in other areas like biologics, peptides, small molecules, really taking the right approach depending upon the disease.
Thank you, Harold, and congratulations to you and the team at BioMarin on your 25th anniversary as a company. Quite an impressive accomplishment. So as we heard from Emile and Janet, 2021 was an impressive year of a variety of accomplishments, as well as some significant ups and downs in the sector. But we're now looking forward to 2022. It's looking like it's going to be shaping up to be the best year ever in terms of approvals of gene therapies targeting rare diseases. Maybe each of you could just share a little bit about your views and perspectives on now, after this period of some success, but also continued challenges. Are we now poised for more consistent growth across the sector, and particularly in this rare disease genetic medicine category? So Suma, why don't we start with you? If you want to share your thoughts.
Absolutely. I think I agree with you, Bob. I think this is very exciting times. 2022 was exciting, but 2023 is going to be even more exciting as we file, I think, the most number of BLAs or applications, both in this specifically in the gene therapy, as you said, cell therapies with CAR-Ts, with the leaders. Now it's the time for gene therapies. Obviously, with previous drugs like Zolgensma and Luxturna as the previous gene therapies. I think it's exciting years with BioMarin and us with Crystal Biotech, with BVAC for epidermolysis bullosa, where we announced a positive data. I think gene therapy is going to be, I think the 2023 is going to be the banner year for gene therapy. So we're really excited. Again, I think with gene therapies, with some of the challenges with the CMC, which we're going to speak later, but also the regulatory pathways for gene therapy, working with the OTAT division and learning all of the changing guidances and keeping up with the requirements specifically for some of the endpoints with different diseases, because as you know, OTAT works across 6,000 different rare diseases in the gene therapy space. And some of the challenges could be, disease-specific areas where FDA or the division has to come up to speed with the specific disease, the endpoints. How do we define endpoints? Or maybe for diseases for us, for epidermolysis bullosa, we have a very fixed endpoint, which is wound closure. But then our other pipeline diseases like ichthyosis or Netherton's disease, these diseases have no defined endpoints. So some of the challenges will be, again, working with the agencies on, defining these endpoints, validating these endpoints, along with the challenges of even getting the gene therapy and administration process ironed out with the division.
Thanks, Suma. Harold, why don't you share your thoughts on the same question?
Sure. Well, so as Janet indicated, we just announced that our Roctavian program for hemophilia A achieved stable and durable annualized bleeding control in probably the largest phase 3 gene therapy study in hemophilia A patients to date. So we're very excited about that. We're able to apply these learnings to our 2 other earlier gene therapy programs, one for PKU, which is a disease that we've been involved with for many years, as well as a new program in hereditary angioedema. I think that the understanding of the science of gene therapy continues to grow for us at BioMarin. We're leveraging our learnings from the Roctavian program for our other programs. I think as clinical development advances, the regulatory framework to support these products also continues to develop. Health authorities will need to keep up with the pace of how quickly the science is evolving. However, we, and I know our colleagues here on the call, want to partner with agencies to establish really clear, reliable development and approval pathways for these products. So, I think we owe this to our patients. So we hope to see more approvals in the coming year, especially at BioMarin. And a lot of this, as Janet and Emile mentioned, are really based on the work that's been done by ARM and our industry colleagues with health authorities to really define the concerns and ways to address this.
Thanks, Harold. Bobby, why don't we turn to you?
Yeah, thank you very much. I mentioned in the introduction that we have an approved product for metachromatic leukodystrophy in the EU, and we're excited to bring that to the US with a filing probably late 2022 or early 2023. But just the difference that this product can make is just extraordinary. So metachromatic leukodystrophy, very rare condition, but it's essentially, and I can see Jason nodding his head because he has a similar story in cerebral adrenoleukodystrophy as well. But they're very similar in that they have, there are no treatment options available for MLD. These children lose the ability to talk, to interact, and unfortunately, many die in the first decade of life. And there's really only palliative care. What we've seen is that a single administration of gene-modified cells given early before symptoms have started or have just started can essentially lead to a relatively normal life for these kids. And we have kids who were treated over 10 years ago with a single administration of gene-modified cells who are now going to school, kind of playing with their friends and everything else. And it is extraordinary what this type of approach can do. And this is a study that started, I say, over 10 years ago. We've got the approval in the EU. And I just think on the regulatory front, we've worked with the FDA, had conversations, we've had patient support groups come and accompany us to those meetings and demonstrate what an impact that this can make. And so we're excited to bring that to the US with a filing, as I said, hopefully towards the end of the year, early 2023. And just the same approach is being used for a condition called Wiskott-Orer syndrome, which is an immune deficiency. And we've just had some interactions with the EU regulators and now are guiding to a filing in mid-2022 for Wiskott-Orer syndrome in Europe, and now have expecting further interactions with the FDA as to when we can bring that to the US. So these are both programs that started over 10 years ago, and so it's a huge amount of work. There's the kind of durability that's been established, but I think it's to the point you're making, it takes a long time to bring these through, to have those interactions, to get the guidance as to what is required, to Suma's point, in order to file. But hopefully, these kind of legacy programs that are now getting to these final stages and being approved will pave the way for more expedited approvals in the future for future programs, because we know that this approach works clinically. The difference it makes is extraordinary, but knowing what is required from the regulators to get them over the line as approved medicines is a different thing, and hopefully we can do that more effectively in the future with the learnings that we have in these early programs.
Thanks, Bobby. Great insights. And why don't we turn to Jason? I know that it looks like he's itching to share his perspective as well.
Sure. No, thanks, Bob. Yeah, so I think 2022 is going to be really a watershed year for our field. And I joke sometimes that, gene therapy is the 20-year overnight success story, right?
Yes.
That, pioneers in the space, and, Bobby's been doing this way longer than I have, really started, the sort of revolution of what these kind of genetic medicines can be. And now we are really starting to see that pay off for patients and for our healthcare system. And I think that's just super exciting. So Bluebird, for our part, right, two of the therapies that we've been working on for, almost a decade, right, are in front of the agency right now. Beti-cel for beta thalassemia and Eli-cel for cerebral adrenoleukodystrophy. It's like a spelling contest to name some of these rare diseases. And I think all of the sort of work, whether it's on the scientific side or the clinical side or the manufacturing side or the market access side, is really, coming to a head right now. And, that slide Janet put up just a few minutes ago, to see that pipeline or set of waves start to break on the shore now is just really exciting. And I'm proud to be not only a part of my company, but a part of this industry to bring this kind of really positive change for the lives of patients and their families.
Thanks, Jason. And really looking forward to seeing, this continued success within the sector, and particularly, following the programs that you have with your team there at Bluebird. Maybe if you could just, we'll just transition a little bit. You had mentioned that some of the regulatory obstacles, maybe you could just punctuate that with an example or two, and then we'll also touch base with Harold and Suma on the same topic.
Yeah, no, I'd be happy to. I think, Bluebird's experience is really part of acknowledging that both the FDA and EMA and all of us gene therapy companies, we're really creating the regulatory framework together, right? So, each product is its own thing, it's its own disease state. And the agency's been incredibly engaged, right? Especially on the clinical side. And we referenced the Rmat designation that ARM was so instrumental in helping make a reality. Bluebird has taken tremendous advantage of Rmat designations to have regular engagement with the agency. But I think the biggest challenge has been on the CMC or the manufacturing space, where, for Bluebird's part, we have 3 programs, in various stages in front of the agency, where we've had to, understand their expectations, right? So, our products are ex vivo products. The FDA has said publicly that these kind of BLAs, it's like 80% CMC and like 20% clinical that they're focused on. We're all familiar with the amazing clinical benefits these kind of products can have. But now what I think both the agency and industry is realizing is the agency wants to set a really high bar in terms of predictability of the manufacturer. Our tech ops people remind me that, the process, your product is the process. That's been true forever. It is acutely true in these advanced, kind of therapies. And so I think as we've been moving along, we're glad that the agency is setting an appropriately high bar for what kind of release controls, what kind of consistency they want to see in manufacturing, what kind of traceability. But I think it took a few years and multiple iterative engagements with the agency to fully understand the standards they want to set and how we as companies working in this space can meet those high standards. And I hope in the future, I think we take the lessons of RMAT and apply it more acutely in the CMC space. So there's just much more engagement, much more understanding around the standards they want to set and apply, not only to products that are getting filed and approved now, but I think somebody mentioned it earlier, for the next wave of products, so we can get these to patients even faster.
Great, thanks, Jason. Thanks for sharing that perspective. Harold, why don't we turn to you to see if there's any unique examples with your experience at BioMarin that would be helpful for our audience to hear about.
Sure. So first, I just want to echo Jason's comment that really, in terms of these regulatory interactions, we don't see them as obstacles, but it's really a process of learning for us and the health authorities that we're working with. From a CMC perspective, BioMarin has been somewhat of a trailblazer for manufacturing, but we recognize this is still evolving, especially as the technology and products evolve. But what I wanted to do was maybe comment a little bit more on the clinical development aspects.
Sure.
And I think for us, the two main issues are safety and then durability. In terms of safety, there are still issues we're learning about. How best to interpret preclinical findings? Will they translate into the clinic? I think we, FDA, patients, clinicians, we all are aligned on the importance of human safety when developing these new treatment platforms. Clearly, there are some defined checkpoints that should be there, such as clinical holds, as onerous as they may be, to really ensure the highest safety standards across all stages of development. I think we've experienced those. We're working through those with the FDA, but they're important. They're important in our responsibility to patients. I think the second thing is, durability, and I think Jason commented on this as well. But it's really, how long-lived will these therapies be? With Roctavian, since that's top of mind for me today. We received a complete response letter, which really introduced a new recommendation for 2 years of data from our phase 3 study. Fortunately, as we mentioned earlier, we now have those data. They're really great looking data, and so we're ready to re-engage with the agency. But, I think questions of durability was one of the reasons that pushed out our discussions with the FDA. We actually, at the same time, engaged the EMA, and we've continued to have discussions with them, over the course of this time. And so we're expecting to be able to bring those to a close this year as well.
Great. Thank you, Harold. And Suma?
I'll expand a little bit on what Jason Harold touched. So I'll go from a clinical perspective. Obviously, with Krystal Bio, we're a little unique because we're going to be the first ever topical gene therapy using an in vivo approach, but with a redosable gene therapy. So quite different from what Harold or Jason, you guys are tackling something which is almost one and done. So durability becomes the key, important. So for us, it's very different from the angle. We have to show repeat administration, immunogenicity, and safety, of course, because it's repeat administration. You want to see that the drug can still be effective, be safe, and still, be efficacious. So this is our barrier that we had to face and show that data. So slightly different from what BioMarin, but again, as I said, is the division is again seeing different kinds of therapies with AAV, lentivirus, HSV. Topical gene therapy was something new for the division. So a little bit of educating and working with them. Again, we're very fortunate we got the RMAT status because we had some strong phase 1 data that enabled us. So helped us with that interaction very earlier on with the division, specifically in a skin disease, with skin being turned over. It's an organ which turns over, unlike other organs where you can the virus can integrate and have long-term efficacy. In our case, because of an organ or the skin with its turnaround, we had to come up with a solution that was redosable and open wounds for these patients where to administer the drug in a painless manner, because these patients have, open epidermolysis bullosa is called one of the worst diseases, because these patients don't produce what it calls collagen 7 protein that holds the skin together. So obviously we can't, invasive approaches can be very challenging. So again, so we had to think creatively, come up with a solution where we could, formulate it as a gel and apply. So as the skin closes, you can correct the skin and get durability of wound healing. So the challenges we faced was twofold. One was, again, efficacy endpoints, because the FDA is very familiar with using guidance documents for chronic wounds or acute wounds. They're 20-year-old guidances, and you're stuck with that. But when you work with epidermolysis bullosa, it's not a disease of wound healing. These patients do this, the wounds do heal, but it is the fragility of the skin. So we had to educate the FDA and how to think differently. It was challenging. But again, we worked past with it. We had some wins and some we had to, make sure we could be consistent with work with the FDA, because obviously I mean, I remember talking to the reviewer, and one of the first things when the meeting was over, she came up to me and she said, you know what, the reason I ask you for these endpoints is because you have to help us, define that label, because you have to give us enough data to give us a label that's meaningful, that can help these patients. So I understand the FDA's challenges too. So you have to work with them, and hopefully, I think we came up with a solution that was a win-win for both of us. So that was in the clinical end. The CMC, again, 2016, we were fortunate when we started the company and had the Rmat status. The OTAT division was not as busy. I mean, they were much more interactive. I mean, in fact, I would have direct communications with the CMC reviewer herself. So we were very fortunate because we got guidance from the very beginning. I think some of the experiences they had with the prior gene therapies, especially with potency assay, we all know that, and we've gone through that. I mean, I think they warned us, they said, make sure you have your potency assay. If you're ready to go to phase 3, if you don't have your potency assay, then you cannot move to a phase 3. So again, helping us with these, information helped us really, put our effort into that and come up with a viable potency assay that is, that tells you what your efficacy will be, or a marker for your efficacy clinical. So these are the things that helped us develop and invest in all of these things. And along the way, what we learned was, outsourcing some of these activities can be very, very challenging. So you have to bring your scientists in-house and focus and be committed. Unlike small molecules or antibodies, which is a very evolved space, outsourcing and going to outside the world and getting all these assays developed is easy, but gene therapy is very, very unique. And if you want it done correctly and in a timely fashion, we have to invest that in-house. So those are the two lessons we learned. Bring your assays in-house, bring manufacturing, understanding scalability, make sure you have and having control of that is very, very, very critical for success.
Thanks, Uma. Excellent examples, and thanks for sharing your experiences. Hey, Bobby, we have a question from the audience that maybe I'll direct to you, and then Jason, Harold, Suma, feel free to chime in. But since we're on the topic of CMC and manufacturing, the question is, what is the best way to find manufacturing quality requirements from the FDA?
I think that there's a very simple answer to that question, is to talk to them early on and talk to them about what you're doing at a regular, in a regular manner. I think it does help with Rmat designation to be able to have more interactions at each step of the way. And one of the things that we learned, certainly as far as CMC is concerned, is that the process, and somebody made the point about the product being the process, because these programs have been so long in evolution, has evolved over time. So the way the lentiviral vector has been manufactured, there have been refinements which could be seen as changes. Similarly, when you transduce the hematopoietic stem cells, there have been refinements to that process as well. Now, of course, any change then requires comparability exercises, etc. So, that is part and parcel of what we've had to do and show comparability between what we started with and our evolutions along the way. And of course, one big thing that we did was also to cryopreserve our product so it would be could be accessible to a wider distribution of patients, and to demonstrate that the cryopreserved product acted in the same way as the fresh formulation. And we have an awful lot of data to be able to demonstrate that. But those have been some of the, as I say, the challenges that have almost naturally occurred as a result of bringing a complex product to the world. And so having engagement with the agency about what you're doing, what needs to be used to demonstrate comparability, I think is extremely important. And for the future learning is, of course, try and start with exactly what you're going to finish with. And that's going to be, again, will make things an awful lot simpler in the future. But, this is a learning, this is how we learn as we encounter or pioneer really a completely new field. So I think, but the simple answer is as much interaction as you can with the agency at an early stage, especially when you're doing something like we all are that has never been done before.
Yeah, excellent points, Bobby. And also just give a big shout out to Janet and the staff at ARM and the member companies that participated in the aging project and the document that kind of speaks to many of these manufacturing issues. And the best ways to address them are through the aging document that anyone can access through the ARM website. So just a shout out to Janet and the team and the great work that many of our member companies participated in preparing that document. And also there's other references available on the FDA website. You could go to the CMC manufacturing guidance documents that Sumo also mentioned. Maybe just to round things out on CMC, Harold, do you want to just give a little bit of insight into some of the issues and challenges and the approach that BioMarin has taken? And then
Sure, you know, I think Bobby captured a lot of the essence of where all of us have been thinking. You know, key elements have been, you know, talking to the health authorities, you know, as we go, really trying to minimize the number of changes in the process along the way, really ensuring that we have robust processes, especially around purity, and being able to anticipate scale up based on the size of the population. We have a gene therapy manufacturing facility that produces 10,000 doses a year. And so I think the other thing is, as we've transitioned from our experience with Roctavian to our other earlier programs that I mentioned, we're really trying to refine the process early on so that what we go into the clinic with is as close to sort of a final market image as technology allows us to have. So, you know, I think those are the main issues. And of course, for AAV-mediated gene therapy, which has been around for a while, you know, that continues to mature. And so we can, you know, we can say, yes, we can scale up. We have this amazing manufacturing facility. Obviously, for new technologies where we're still learning in the clinic, you know, based on clinical experience about how maybe we have to modify, it's going to be a little different as it evolves.
Yeah, fully agree, Harold. Jason, since you're in the ex vivo approach, why don't you share a little bit about your insights on CMC challenges?
Yeah, I think it's, you know, Bobby touched on this a bit earlier. You know, Bluebird was my first cell-based medicine company. I came from the world of small molecules before, and it's actually a blessing to be able to refine your product in the midst of clinical trials, right? The reason you do clinical research is to learn and then apply those learnings. And so with Bluebird's programs, you know, we had the opportunity to get data from real patients that had us change our processes and product along the way, right? Whether that's starting material, whether that's transduction protocols, you know, how we make the vector, all of that. But then, of course, eventually the agency is like, so can I get some comparability data for everything you did over the past few years to get there? And I think for me, that's a bit of kind of paying the scientific debt of starting as a pioneer, learning in the clinic, and then getting to the point where you have a tight process with all these lovely potency assays that they want us to do, that then allows it to make it into the hands of healthcare providers and patients. So, you know, there are days at work where I bemoan the learnings and how to have to apply them, you know, in a new timeline or in a new context. But I'm an optimist, and I believe all these CMC learnings, I think, as Harold said, allow us to start in a better spot, right, with a set of processes and approaches that allow us to run these studies and move forward and, you know, ask a different set of medical questions that hopefully we can answer, you know, in a more efficient way.
Excellent points. Thanks, Jason. Maybe on the topic of learning and as we evolve from research to clinical development, obviously empowered by capable manufacturing infrastructure and processes, onto kind of the next step in the evolution of products once you get regulatory filings, and each of your companies are either in the process of doing that and hopefully soon will be approved. We're all rooting for you collectively across the sector. You know, the next big challenge, and maybe this is an area of also pioneering work, is commercialization. And, you know, some of the early gene therapy products have had some challenges and maybe even some disappointments along the way. But Bobby, we'll start with you. And what do you see as kind of the key learnings from the first wave of gene therapies, and how are you and each of our collective companies within the sector are applying those learnings to enable greater successes with your soon to be approved products.
Yeah, no, that's a great point, because, you know, we've talked about the regulatory kind of milestone, but then there's the commercial milestone as well. And so that's you know, and that when I talk to our staff, I talk about the fact that we're pioneering almost at every step of the way, whether it's a scientific development, clinical development, regulatory, manufacturing, but commercial is also a pioneering step as well, because, you know, the systems aren't ready for, or are certainly, you know, they're certainly not ready for once-off therapies. They're designed for something completely different. So, you know, that is in itself another pioneering element that we are going to. And in our case, I think, you know, with what we've learned from Limeldi for MLD, it's really about the, you know, about the indication, you know, about the severity of the disease that we're treating, the severe unmet need, and the difference that we can make with a once-off gene therapy. And the magnitude of difference is just so extraordinary that it has been, I have to say, you know, when we just talked about our commercialization of it this morning, it has been extremely well received by the reimbursement agencies. We've had some very positive conversations, and I hope, you know, what we'll be able to demonstrate is that they do recognize the value of this therapy, and it will be appropriately reimbursed, which is extremely encouraging for the field. And I know from Jason that experience wasn't there, you know, previously for Zynteglo, which obviously is unfortunate. But I think sometimes it is about the indication and the difference, and that's what we've been able to show with Limeldi. And I think so that again, you know, is a learning. It's a learning for us. It's a learning for, you know, the new products that we are developing, and to ensure that we articulate the value and the difference it makes over the current standard of care or the natural history as effectively as possible in order to realize the true value of the treatment.
Thanks, Bobby. Jason, do you want to share some of your insights and experiences from the Bluebird perspective?
Sure, you know, I'd be happy to. You know, I think the lessons I think Bluebird's learned, some of them have been sort of through our own experience, but others have been from just observing other, let's call it pioneers, you know, in the space, right? So you think about the CAR Ts and some of the initial challenges they had getting, you know, access really at the treatment center level, or, you know, some of the pricing debates that happened around some of the first AAV-based therapies, or our own experiences in Europe. I think what we've taken from all of those learnings, our own or from the space, is really the need to engage early and often across not only the payer types, right, the different, you know, whether they're government or private, but also to not be shy to push policy change. And I think this is where ARM has done a really good job sort of voicing some of the issues for the industry on behalf of all of us. To sort of amplify the voices of each little company that, you know, we all have our own diseases and technology platforms. And so for us, you know, we have been engaging with the commercial side for years now. And that engagement coupled with policy work has really started to show some positive impacts. So just to give an example, last year, CMS implemented this final rule on value-based payment. And this is a rule that had been sort of developed in the prior administration and then continued into the Biden administration. And the consistent engagement by multiple groups, including ARM, got us to a point where we now have much more flexibility to implement creative reimbursement models, both in the private sphere and with government payers like Medicaid and Medicare. And we found that payers remain really engaged and interested in these payment models for all these gene therapies that are going to come to market in the next few years. And I think one thing we've learned, again, probably from our European experience, is to be flexible, right? I think Bobby's point about indication matters. Indication does matter. Payer type matters, right? You know, if you are a state like Georgia, you have a pretty large Medicaid sickle cell population. And so you're going to be much more interested in perhaps a flexible payment model than others might be for a very rare disease like our ALD program, for example. So we're excited to apply our learnings this year and next as we launch a couple of therapies. But I do think I very much agree with Bobby's point that the pioneering and innovation does not stop at the lab or in the clinic. We got to just keep doing it all the way across the spectrum.
You know, it was nice that you mentioned there, Jason, the great work that Janet and Paige and the team at ARM are doing in the policy front. I know that, you know, we're all very supportive of their efforts and, you know, are starting to see some of those efforts turn into real meaningful changes on the policy front, but, you know, more work to be done. Suma, we just probably have about a minute or two left. If I'll give you the last word here, if you want to just wrap up with maybe sharing some of your thinking as you're close to being having an approved product and potentially commercializing.
Again, for us, it's going to be brand new. Again, we being a young company, we've never commercialized anything. So we have a commercial team now. Again, from what I speak with them and what I hear from them is a lot of disease education. Epidermolysis bullosa is such a rare disease and many of the payers have never even heard of the disease. And you would describe it one of the worst diseases because these patients, the kids are born with missing skin. Can you imagine? I mean, they have wounds or open skin throughout the life. And the life expectancy of these patients are on average 30 years of age, and they die. And there is no current therapies or anything available for these patients. So it's basically bandaging and wrapping, and which costs $400,000 a year, $300 to $350. So it's already expensive for these patients. So I think for us, it comes from a very different angle. So when we have a therapy that could, you know, improve and maybe cut cost on some of that, there's a true benefit. So based on our initial discussion with payers and educating of the disease, I had some of these payers, literally tears come down the eyes when you show the pictures of these babies with missing skin. So it's very nice to see the empathy from them. And they said, oh my God, this is a really bad disease and we want to help. So that's our initial reaction. So it's very promising. So I hope, you know, from Jason's experience and Bobby's experiences, I mean, we'll have the same experience and hopefully we will engage ARM and others to help us through this journey of how we can get this product, if it's approved, to these patients where right now, you know, there's nothing for them and it's a pretty bad disease, which they are born with it and they rapidly deteriorate. By age 10 or 15, they lose their ability to use their fingers. The fingers get fused, the legs, they are in wheelchairs. So, and these are very healthy, normal patients. So from from a perspective of, you know, their normalcy with, you know, they're smart and they have all the other abilities and functions, but losing, you know, their hands and legs and watching that rapidly deteriorate is pretty, you know, it's a horrible disease.
Well, yeah, thanks, Suma. I mean, we certainly know that, you know, those patients are in desperate need for an effective and transformational therapy. So we're very much rooting for you and your team at Crystal to have some significant success as you go through the regulatory approval process and get ready for commercialization. I think we're at our time, so I'd just like to thank you, Suma, Bobby, Jason, and Harold for sharing your insights, for taking time out of your busy days. And we wish you much continued success as you march down your different paths toward regulatory approvals and hopefully commercialization in 2022. With that, I think we'll wrap up this section. We'll turn it back to Janet.
Thank you very
Thanks,
much.
Bob. Thanks, everyone, for a terrific conversation and for all you're doing on behalf of the broader cell and gene therapy sector. I now have the pleasure of introducing the moderator of our next panel, whose image I think will be appearing soon. There he is. Hey, Robert. Dr. Robert Engs, CEO of Vore Biopharma, is, I'm happy to say, an active ARM member. Robert has more than 15 years experience in the industry in a variety of roles, including as a senior biotech executive, as I said, currently a CEO, also as a physician, a venture capitalist, and as a strategy consultant. So we're really pleased to have him as the moderator of this next panel. And with that, I will turn it over to him to introduce his panel and his panelists.
Thank you very much, Janet. It's wonderful to speak to everyone. I'm Robert. So just a little bit about Vore Bio. We are a company working on hematopoietic stem cells and CAR T cells in the treatment of cancer. That being said, you're not here to hear me, but you're here to hear from the panelists. And so we have a great panel lined up. This is focused on oncology, but certainly cell therapies do go beyond oncology. Let me firstly have Alison Moore introduce herself from Allogene.
Thanks, Robert. Good morning, good afternoon, everyone. My name is Alison Moore. I'm the Chief Technical Officer at Allogene. It's a pleasure to be here today. My background is by education in cell biology. I have spent time working at Genentech and at Amgen, and most of my career has been in the CMC sciences, the majority of which has been in process development, but also oversight of manufacturing, oversight of supply chain, etc. It's a pleasure to be here today.
Great. So secondly, we'll have Lalo Flores introduce himself from Century.
Hello, Robert. Just to let you know, I mean, the system doesn't let me activate my video, so maybe the host doesn't let me activate it. So it's not that I'm trying to not to use it, but it's something with oh, there it is. Now you're letting me.
All right.
Hello, everyone. Yeah, thank you for the invitation. A pleasure to be here. I mean, Lalo Flores here. Osvaldo is my official name, but go by Lalo. As Century CEO, our company is developing iPSC-based allogeneic cell therapies for cancer. We're only 3 years young. We started the company in the fall of 2018, and we've been very busy building what we think is a next generation platform where we incorporate, state-of-the-art cellular engineering and manufacturing to execute on our mission. We are, about over 160 employees now. We're, headquartered in Philadelphia, but also have research sites in Seattle and Hamilton, Canada, and now more recently in Branchburg, New Jersey, where our manufacturing facility will be based. And yeah, so a pleasure to be here.
Great, and Lalo, congratulations on the deal announced today with BMS. I'm sure we can hear a bit more about that later in the panel. And not to be outdone, Stephen Kelly from Charisma did a deal today announced with Moderna. Congratulations on that. Would you like to introduce yourself?
Hi, thanks, Robert. Steve Kelly, CEO of Charisma. Charisma is a private Philly-based company. It's a Penn spinout. We're working on engineered macrophages. It's a cell type that up till recently has been sort of under the radar. And I think that over the last several years, as we've been building out our platform and capabilities, I think that more attention will be drawn towards macrophages. And as you say, we were able to attract a good investor for a deal today, or not an investor, but a collaborator in the deal today. But a real pleasure to be here and appreciate the invitation.
Wonderful. So what we're going to try to do on this panel is tackle a few interesting topics, hopefully interesting for those listening in. We are going to tackle some of the safety issues that we all face in the cell therapy space, thinking about different features and engineering we can put into these cells and how we should be thoughtful around that. We'll touch on the advances we're making in the clinical and regulatory space as cell therapies are becoming more commonplace in clinical practice. But why don't we launch on a topic which I think is quite pertinent to the panelists collected here, which is around modalities. Obviously, the original innovation in cell therapy was with autologous CAR T therapies, which have made phenomenal advancement in the field. As we've heard, Gilead and BMS are now pushing into second line, soon to be first line approaches in different cancers. And on top of that, this panel represents now a plethora of emerging cell modality platforms, including things like gamma delta T cells, macrophages, NK cells, HSCs, allo off-the-shelf approaches. So I'd love to get each panelist's thoughts around what the future holds particularly if there's going to be distinct roles for different modalities, or whether one modality might rule them all. So why don't we have Lalo lead us off?
Yeah, thank you for that. Great question, Robert. Yeah, look, it is, I think all of us, I mean, you've seen since 2017, many, several dozen, I don't know, many companies emerge on the cell therapy, being, really inspired by the great promise that autologous CAR-T has shown. And so part of that, was inevitable to see that the desire to move first into allogeneic, go from patient-derived cells to healthy donor-derived, and then in our case, to move even beyond that, starting with iPSC, stem cell-derived cells. But part of that, as we switch, on one hand, the other natural drive was to start thinking, moving beyond immune effector cells such as alpha beta T cells, where most of the clinical data comes from. And so our view at Century, we thought, and we have seen early promising clinical data for NK cells, the other major, as you know, immune effector cell of the innate system, come with some intriguing clinical validation. And now we are also advancing, and there are other companies also joining the effort of pursuing gamma delta. Steve will talk about macrophages, but our thinking is that there is all of these various platforms. There's an opportunity that it's most likely they will coexist. Certainly our approach at Century is to really leverage the different biology. NK cells have a very different biology than alpha beta T cells, right? They don't expand upon recognition of antigen the way an alpha beta T cell will do. They don't have that traditional memory phenotype. And that's what has made alpha beta T cells ideal for this one and done approach, of the early autologous. But now we do things that, NK cells, complement, if you will, what alpha betas can do and gamma delta T cells can do, in that they have a different biology, right? They don't secrete all of these pro-inflammatory cytokines upon activation like alpha beta cells, and therefore we're seeing a much better safety profiles, which we think is an advantage of NK cells. Also, they have the fact that they have shorter, they don't expand so much, they don't persist as long as these cells. We think they will have advantages in certain settings where, like AML, for example, is a good example where even the best targets, there can be found not just on cancer cells, but also in normal myeloid tissue, and creates a challenge in terms of therapeutic window where we can take advantage of the shorter half-life of NK cells to build greater, safety windows, right? So this is just one example. We see that, these cells, modalities could potentially be used in combination in the future, where an NK cell could be used, therapy could be used to debulk, tumors, and so then which could be followed by a T cell approach, right? That it would be safer, and so on. So we think, bottom line, the answer to your question is that I think that a lot of these different platforms will coexist. There's also, we're excited to explore the benefits of even combinations in between products derived from different platforms. So I'll stop there.
Thank you, Lalo. That's great. Maybe Alison, would you like to add on to that? You're also working on iPSC-derived cells, and Allogene is obviously one of the pioneers in the allogeneic off-the-shelf space.
Yeah, thank you. So Allogene is entirely focused on allogeneic biology, but will certainly evaluate and investigate, hopefully execute whatever levers might help get us there. Our position is, in terms of your broader question about modalities, I think what we ourselves and many colleagues in the field have established is that deletion of the T cell receptor affords us control over graft versus host disease. And then there are, of course, multiple different approaches for platforms in overseeing the other immune components of allogeneic biology. Allogene happens to use a CD52 knockout in combination with an anti-CD52 monoclonal antibody. But there are obviously different ways that one could approach lymphodepletion or stealth approaches in the future, and we're seeing some of those being advanced, and we're advancing those ourselves. We also have a collaboration with Notch Therapeutics, as you mentioned, Robert, in the iPSC space. We agree that that will be important for the future of our field. And we also agree with Lalo in that we think that the power of these immune cells, a lot of the validation in the heme space, and then progression into solid tumor space will open up, plenty of opportunity for many of these modalities to coexist.
Thank you, Alison. Stephen, I'd love to hear a bit more about your macrophage approach and your general thoughts on the plethora of modalities.
Absolutely. Yeah, so as Alison Lalo said, there are a variety of cell types under development. All of them have distinct functionality. There are different approaches: the autologous approach, the allogeneic approach, there's in vivo approaches that are emerging. And I think another area that I think about is integration into patient care. What's the best way to do this? Whether it's a completely cell therapy approach, or we combine with a T-cell checkpoint inhibitor or something like that. So there's a lot to your question. Opens up a lot of directions on how to answer. With us, we're obviously currently an autologous CAR macrophage company. We think we have distinct advantages in certain areas in solid tumors. We can traffic to cells, we can to tumors, we can worm the TME, we can attract other cell types in, like T cells, NK cells, et cetera. Which I think goes to the last question, and I think that both Lalo and Alison were talking about. There is going to be this integration. And so we're doing a collaboration right now with a CAR macrophage plus a CAR T. We can get into the tumor, we can worm the TME, remove the immunosuppressive signal, and recruit T cells in. That sets it up very well for something that's such an exquisite killer like a CAR T. And so I think you're going to see those integration strategies. At the end of the day, we have an immune system with distinct cell types. And I think that as time goes on, they're going to be integrated in a therapeutic approach. We're excited to have one element of it, we're looking forward to working with other companies in the area as well.
Thanks very much. I totally agree with that. At Vore, we're working on both hematopoietic stem cells and CARs, and we think there really is a synergy to exploit the biology and create great therapies for patients. So let's pivot towards a current topic, particularly highlighted recently by your company, Alison, which is not an old topic, but certainly it's come to the fore around safety, and that we're manipulating cells, and some of us are using gene editing strategies, and there's always the fear that you could potentially generate something with oncogenic potential, I know you may be limited, Alison, but if you could just open with comments here about how concerned the field should be around potential safety issues.
Yeah, thank you, Robert. So as many folks will know, a few months ago, Allogene was placed on a clinical hold because of the observation of a chromosomal abnormality after administration of our product. Actually, today, the FDA has released our hold across all INDs, so very happy about that.
Congratulations.
Thank you. And I think, first of all, we fully appreciate the collaboration of the FDA and their upholding of their mission to protect patient and public safety. And we are all working in a field that is highly emergent, and we're doing explorative work. And so we all have a responsibility to be extraordinarily vigilant and to take appropriate steps when necessary. I think more specifically with respect to gene editing, I guess this comment was relevant to the last question around modalities also. Currently, we are editing 2 genes in our platform, TCR gene and the CD52 gene. But obviously the possibilities there are numerous, and folks are designing more and more sophisticated ways in the ex vivo setting. And of course, we'll see this translate into the in vivo setting to create gene manipulations. I think the unintended consequences, if there are any of those gene manipulations, are being studied in parallel. And we can study lots of them in vitro, but where the rubber meets the road is in vivo. And speaking from my discipline on the CMC side of the house, we have a huge responsibility. I mean, CMC's mission across all companies is to demonstrate to regulatory bodies that we can reproducibly produce safe and effective product. So it's really the definition of safety in the gene editing space that's not a single or fixed definition. That definition is evolving as we learn, all of us collectively, about our products. And ultimately, as everyone knows, if we aspire to commercialize a product, we have to demonstrate a control strategy that is indeed in control. So there has been super intense learning for Allogene in this hold period of time. We've done some amazing biology, and we won't be publishing that information. But hopefully many of our industry colleagues, as we proceed forward here, will be sharing all kinds of information that will pave the way for the future of medicine with highly manipulated and genetically engineered drugs.
Thanks, Alison. Steve, my understanding is you're using AAV, which is, by its nature, a directed insertion approach, but love to hear the reason behind that, if that was motivated by safety reasons.
Yeah, actually, we're using an adenoviral vector, so it's a non-integrated vector. So it's we are maybe a little bit different on the panel. We're as you know, integrating vectors have the potential to really have insertional mutagenesis and potentially oncogenesis. I think it's a rare issue and decreases as the cells become more differentiated. We're fortunate, we're using a non-integrating vector with an adenovirus. We are using or focused on macrophages which are terminally differentiated. So it's a low level risk for us, but certainly safety is paramount. And as we explore the development, unlike the T cell history and NK cell history, we have rather than 20 years and 10 years respectively, we have 6 months of clinical history. So safety is a key issue for us and what we're looking at.
Great. Lalo, any additional comments?
Yeah, absolutely. So look, first of all, I think that we all think and we're confident that all theoretical and real safety risks that may come from genetic modifications are addressable and manageable. And we need time. We're innovating at such a rapid pace, so that some guidelines need to be developed. But we're very confident. We have the analytical tools, the processes in place to manage those. But let me just say one thing in addition, that what I believe is, the reason why allogeneic cell therapy with engineered cells has such an incredible disruptive potential is because we are bringing together that concept of using living drugs that can be genetically modified, so we can have the right functionality. We can train these cells to avoid immunosuppressive signals on the tumor microenvironment. We can allow them to secrete potentially, I'm talking about theoretically the dream. This is what we're dreaming, right? About the technology, right? To secrete, drugs or other immune effector molecules that can modulate the host immune system and do more than one thing, things that an antibody drug could never match, right? So this is the disruptive potential. And to do that, to realize that vision, we need to incorporate genetic modification ex vivo to these cells in a safe fashion. And there's no question in our mind at Century that in collaboration with regulatory agencies, we'll be able to do that. And as I said, there are, one advantage of our platform, iPSC, over donor derives as we're all aware, is that, we have the benefit to be able to incorporate our gene edits in a sequential fashion. If you multiplex, the more you try to do in one step, the greater the number of potential, unwanted modifications you can incorporate into your product. So doing this sequentially helps. But finally, if you can select a single clone where you screen for these unwanted modifications and select them out, so you can, start your drug, manufacturing process with a master cell bank of a well-characterized clone. That's one approach where you can really overcome this risk, right? And the other one, of course, look, we need to keep in mind that the agency knows this, and everybody knows this, that there are different types of abnormalities. Some of them are introduced as a side effect of nuclease editing, right? But others are a function of cell division. They're normal, and some of them exist in our donors, right? And so there are different risk profiles. So that's something else that we need to keep in mind and generate the right data. Not all of these modifications have the same type of risk profile. And so I'm very optimistic that we will overcome all of this. Is because we have the right processes, technologies, and analytical tools to really look at this. But certainly, I think if we're going to get to talk about regulatory landscape, is something that we see that there's a tremendous opportunity for the industry to collaborate and have an active dialogue with the regulatory agencies on how to think about this.
Fantastic, thank you. And I second the congratulations, Alison, on the lifting of the clinical hold. It also sounds like Allogene did some fantastic work on that assay development side to make sure that you're developing safe medicines for patients. So that's awesome. Why don't we move on to a next question, which is really around engineering cells. So there's all kinds of bells and whistles and features that we'd like to see, thinking about controlling activity in vivo of these cells, the in vivo expansion, hiding these cells becoming hypoimmune to the host immune system, the manipulating phenotype. I'd love the panel's just general comments on this. What features are important? And how do we know they're important in terms of the success factors we should be looking for, both in vitro, in animals and in the clinic? Lalo, would you like to open on this one?
Yeah, absolutely. Yeah, I'd be happy to. Look, this is what I was saying earlier. This is what is really exciting about our field right now, that we really have the unique opportunity to innovate, where there are just so many possibilities enabled by bringing together gene editing with living drugs, right, with cell therapy. And so, for example, in our first product, Century 101, that will be entering the clinic in the next few months, we have 6 gene edits. 3 of those are designed to allow our cells to resist rejection. We think that that's a major technical barrier and for allogeneic therapy in general, right? So those we think are critical edits that most allogeneic companies will be incorporating it somewhere or another, or different versions of them, to block the major pathways of rejection: CD8 T cells, CD4 T cells, NK cells, in our case, down the road macrophages, right? And humoral rejection as well. So these are all I think key modifications that most allogeneic therapies will need to have, and certainly something that we're pushing hard at Century. But then we know that persistence is important, of course, and so if we can eliminate lymphodepletion regimens, but then allow ourselves to have enough homeostatic cytokine support, we can engineer those cytokines into our cell products. Another modification that we have incorporated, like others, is the production of IL-15, for example, a major homeostatic cytokines for NK cells and also T cells. But in the future, we can add the other ones, right? So that's another example of a natural. And then there is a third category that, you know, many companies would be doing. That is, you know, incorporating engineering strategies that allow the cells to overcome immunosuppressive signals, right? And so when we start thinking about solid tumors, you know, that's going to be, you're going to see a lot of other, you know, modifications aiming at that. And then, you know, as I was saying earlier, we can also engineer, you'll see engineering strategies in which these cells will also be secreting other drugs. You know, you can think of them as micropharmacies, where we may be able to engineer them to secrete an anti-PD-1, right? Or some other antibody therapeutic. Or another immune effector cells to go ahead and activate, you know, certain subset of cells, right? To boost the, you know, to do a two-pronged approach in solid tumors, right? You're incoming, you know, allogeneic therapy and also reactivating the TLs in the tumor microenvironment. So, you know, this is what is so exciting about allogeneic cell therapy, and that's what we have to overcome and learn how to incorporate these gene edits in a safe fashion. But there are many opportunities. I think pointed to one of the obvious ones.
Thank you, Lalo. Alison, would you like to further these comments?
Well, I think it was good to start with Lalo because he's starting with iPSC. So in theory, you can almost go anywhere. So I guess in the field, you know, those of us who are starting with donor material achieve, you know, different phenotypes through separation and culture conditions, or starting with iPSCs through differentiation. So really the only limit, you know, we're limited by our own imagination, to use that cliche, right? Because the force behind the biology in the field here, I think what we're trying to do individually is technically difficult at this moment in time, but the collective force in terms of the progression here is quite enormous. So with respect to, you know, features, Allogene has been excited to have our TurboCAR variant enter the clinic last year. And we're super excited to have that trial restart now. So as Lalo mentioned, that's a modification with a constitutively active modified interleukin receptor. But the possibilities of how we might manipulate cells looking forward is almost endless. And so, you know, one of the most difficult things I think in any of our companies is actually prioritizing where we choose to spend our time. So at Allogene, you know, our research organization is highly productive and plugged in, and we have many advanced opportunities coming in our pipeline. The question is, you know, what do we choose to advance? And then also a little bit more tactically, you know, for us to really advance those opportunities, it's not just about what is the what's the protein expression, what's the phenotype of the cell, what's the genetic manipulation, but it's also about the technology that we use to do that. So of course, many of us are transducing cells with viral vector. You know, many of us are using nucleases for excision or editing, different kinds of nucleases. I mean, that field just continues to advance as well. And then site-specific integration, of course, is a critical technology to enable some of the things that we can imagine. So this is actually, it's a really heterogeneous technology advancement. So that's another reason why we have to first and foremost make our best biological prioritization decisions about what's right for each of our companies in terms of proceeding forward. But then beyond that, we have to really think about, okay, then what are the then consequent technical decisions of what we will use from this beautiful portfolio of technologies that are evolving here. So I feel like, you know, ourselves and those in the panel before us are pioneering areas, you know, over now, for example, like BioMarin over decades, but I still feel like we're only scratching the surface. And I think my colleagues might agree, because you know, once we're discovering, oh, if we can do this, then we can do that, right? That's what this field feels like on a daily basis.
Thank you. Steve, love any final comments? I'm guessing you chose macrophages somewhat because of phenotype and that you could avoid certain edits to manipulate those cells, but love to get your thoughts here.
Yeah, absolutely. And we're fortunate. All the peers, the pioneers before us that Alison spoke of is you know, we can leverage those tools. So all the adoptive cell therapy tools that have been generated over the last 20 years are very useful to us. We're breaking new ground with macrophages. But you know, we've made over 200 CAR constructs for our CAR macrophages. We know each element of the CAR construct itself is important, whether it's the binding site, binding affinity, hinge length, signaling domains, all those things are important. And you know, you have to go through this exercise of what works well for each particular target and each particular cell type. So even we're early on in that, and so we're trying to think of high throughput ways to engineer and tailor each one of those elements. There are things for us. Phenotype of a macrophage is important. Do you want to have a pro-inflammatory cell? Do you have an immunosuppressive cell? And we can tune the phenotype of that as well. I think that we're fortunate that macrophages can traffic to tumors, but we've heard of other cell therapy players that are looking at chemokines and chemokine receptors. How can we enhance the ability for cells to get to the location that they want? And then similarly, expressing chemokines to attract other cells in. So there's a lot of things you can do that. You can change the environment with other transgenes for cytokines to enhance the potential of those particular cell types. You can do gene editing. We're looking at CRISPR editing of SIRP alpha, so blocking the CD47 SIRP alpha access from the macrophage site. So there's so many tools available to us, and I think that my other panelists expressed that we're at the early stages, and there's a lot of learning left to go forward here. And again, we're fortunate that there have been pioneers ahead of us in adoptive cell therapy, and we can apply those and continue to be pioneers in monocytes and macrophages.
Thanks. So let's round off with a question on, really an open question on clinical and regulatory. Now, we're in such an interesting point where now Gilead and BMS exploring, you know, label expansion opportunities for cell therapies, and there's probably around 400 programs that FDA is regulating in the clinic in the US. We know that cell therapies are very unique in the way they need to be developed, and regulators have adapted somewhat to those unique ways. But what does the panel think are the key challenges and opportunities that we face from a regulatory and clinical perspective? Maybe Steve, do you want to lead us off?
Yeah, I think that this is, to use Lala's words, these are living drugs, and we are making changes, learning on the fly. And I think that we have to have, or we're fortunate that the FDA, I believe, is really along for the ride, and they're putting together you know, draft guidance and how would they regulate cell and gene therapy, and how and really embracing the industry. How would we go after these things? And so in September, there was draft guidance from the FDA about, okay, if you're going to make changes in X, Y, or Z, these could fall under the same IND. You know, it's really looking at ways to develop a safe and efficacious approach. And for us, we're early, we're phase 1, and there's a lot of ground to cover, and you know, I think maybe the other panelists have a little bit more insight in terms of, you know, specifically what do they have to overcome. But I think we've been fortunate to have a very helpful FDA in our process as we develop our drugs.
Thank you, Steve. Alison.
Maybe I'll talk more specifically about CMC regulatory. I wanted to pick up on something from the prior panel, the classic phrase that the process defines the product. Of course, that is sort of a practical reality. I don't prefer this phrase though, because as Steve just said, we're working in a field that is just really emergent and dynamic. We all know, even on the fastest, most accelerated path, it takes years to develop a safe and effective product. And in those years, you know, there will be so many advances in the field. So what I love about the example from autologous T-cell therapy that you started with, Robert, in this question, is that those colleagues have the privilege of having demonstrated a safe and effective product. And now they're able to mobilize their resources to say, how can we best bring this to the patient populations that can benefit from it, right? Before you get to that point, you know, we're all busy working away towards achieving that same privilege, right? But as Allogene's recent experience showed, and to your comment, Robert, the dynamics of this field are not just about what are the new bells and whistles that we can attach to the product, but also how do we describe the product? How do we understand the product? How do we characterize the product? And new methods. I mean, the pace of change in even the analytical space is incredible. That's a wonderful thing for us. So actually, the CMC space is very dynamic. And what our regulators want is, it sounds, you know, easy, a well-characterized biologic. And also, as I mentioned in a prior comment, a control strategy. Can you defend that the way that you routinely manufacture your product is happening under a state of control that can be described? So I want to be able to take advantage of all of those measurement opportunities. And I think that we can all agree that in the cell and gene therapy space in general, right, we don't yet have the skill set, the capability to describe a full portfolio of critical quality attributes. So our regulators, in the absence of that, are working really, really hard with us to help us move the whole field towards being able to do that. So in the meantime, though, you know, as many have mentioned prior to me, you know, they are learning alongside us. So on the regulatory front, these drugs that we're all developing, these living drugs, have the potential to be revolutionary. But on the CMC side, they must be shown to be safe, effective, and reproducibly made. And that's why we're getting a little bit out of whack sometimes with, you know, sometimes jaw-dropping clinical data, but being held up or perceived to be held up by CMC items. So it's actually, you know, a fascinating field to practice in the CMC sciences these days.
Thanks, Alison. Any last comments, Lalo? And I would like to squeeze in one more tiny question on partnerships, which is current.
Yeah, real quick. I just, you know, I think everything has been said. The only thing I would say just is that, you know, part of the challenges that we have is thanks to this, you know, extremely fast pace of innovation that we're witnessing, right? And so, you know, we're in a situation where we are you know, we have to innovate and bring products forward without having regulators having enough knowledge, experience, time, bandwidth to create appropriate guidelines. So that's a challenge, right? We have to keep moving forward in a way, guessing or without the guidelines in place. But the opportunity at the same time is that we have opportunity to establish productive dialogue with regulatory agencies early on, and the agencies, I think, as other panelists refer, I mean, they are being very collaborative. They have created mechanisms where we can engage earlier and earlier, like the interact mechanism, extremely useful. We need to also, as sponsors, industry companies, we should come together also and exchange lessons learned and suggest white papers and other tools of best practices on CMC. Of course, there are different challenges for companies pursuing donor-derived, like you are, Alison, in Allogene, or Steve, versus what we're doing at IPS is quite different. But nonetheless, there's a lot of commonality, and whatever those are, we should come together as in the industry as well, and try to put our best thinking together. So I think there's a lot of opportunities out there as well to improve on the regulatory front.
Thank you. All right, let's try to do one last question. Only 1 minute each. So the question is, how important are partnerships in this space? Kind of a bit of a biased question, given the recent news from both Steve and Lalo. But Steve, you want to go first?
Absolutely. Well, so partnerships are oftentimes you can get capital from them, but I think importantly, you can also share technology. For us, our deal that we announced with Moderna today brings two pioneers together. We have engineered macrophages, they have mRNA and LNP. It enables us to create a new therapeutic modality, which is in vivo engineered macrophages. So it's fantastic merging of two distinct technologies, and that's what's really important. We had a similar deal announced last month where we're working with Merck on a T-cell checkpoint inhibitor, their approach combined with a macrophage to enhance the overall activity of both mechanisms. So I think partnerships are essential, especially for small companies like Carisma, but I think even larger companies could benefit from that as well.
Thank you. Lalo?
Yeah, no, I completely agree. Partnerships are really important for many reasons. And one of them is, as Steve alluded, is that big pharma partners bring tremendous expertise, not just not to mention technologies, right? But also scientific expertise. I mean, people forget that each individual indication is a completely different set of scientific challenges, and the pathophysiology is different. It's a lot of scientific understanding that you need to develop to execute successful clinical programs. And so having, in our case, multiple myeloma is an example of an indication that we thought was, something that it was too challenging for us to tackle. We already decided to go after two challenging indications and become experts on B-cell malignancies and glioblastoma. We were not going to work on multiple myeloma, for example, on our own. It's very different. So bringing a leader like BMS to be a partner, it makes tremendous sense. The risks, the technical risk, if you will, of those programs. The same thing for AML. AML is a very challenging disease, high risk, high reward. And bringing a company that has deep expertise really is extremely powerful, helpful. And not to mention that also adds you to your, financially makes you stronger, but also from a technical perspective, right? Joining two teams together with complementary, skill sets and technologies, it just makes us stronger.
Thank you, Alison. And Allogene was based on, a founding partner with Pfizer, correct?
That's correct. And just to build on what both of my colleagues have just said, I mean, it is the convergence of disciplines and skills that help us address our lofty aspirations and tackle our challenges. And the convergence also of those experience sets, small companies, large companies that are partners, and in times of achievement, they share in those achievements, and in times of challenge, healthy partnerships can offer, tremendous guidance and support. So we have many partnerships and we appreciate them all.
Thank you. With that, I would really love to thank our panelists, Alison, Lalo, and Steve. Wonderful, very exciting times for us. And I will hand back to Janet.
Thanks, Robert. And thanks, Alison, Steve, and Lalo for joining us on a day when you're making big news in other ways too. So we're really grateful for your time and your thoughts. Thanks everyone for joining us today. I hope this session has given you a good feel for the science, the speed, the scope, and the scale of the regenerative medicine sector, the new paradigms that we are entering thanks to the incredible accomplishments of 2021. We are certainly excited to join with these thought leaders you've heard from today to work collectively to try to advance the policies that are needed and the collaborations that are needed to move the whole field forward. If you would like more information about how to do that in conjunction with us, please feel free to reach out. We would be happy to talk to you about that. With that, I'll thank you again for joining us. Wish you a satisfying and rewarding JP Morgan week. Thanks to our hosts at Biotech Showcase. Take care.